In angiosperms, the endosperm is a nutritive tissue which is triploid in condition. So, this is providing nutrition to the developing structures. This nutritive tissue, or we can say that embryo which is present along with the endosperm inside the seed. So, this endosperm is providing the food materials to the developing embryo inside the seed. So, by the time the seed get matured, if endosperm is retained, or sometimes the endosperm will be completely utilized. So, based on that one we can say that non-endospermic seeds and the endospermic seeds. But how does this endosperm is developed, or how does the endosperm is formed? As we already said that a primary endosperm nucleus, so will be converted into endosperm. In this process, the primary endosperm nucleus will first undergo free nuclear divisions. So, free nuclear division means only the nucleus will divide while the cell does not divide. So, that cell remained only one, whereas the number of nucleus will increases. So, that means consider this is a primary endosperm cell. So, this inside which the number of nuclei will increases without increasing the number of cells, like this, and we call this as a free nuclear division. After some time, what happens? The cellularization will start. Cellularization means each of the nucleus will start collecting some cytoplasmic structure and convert it into cells like this. So, this is a way how the cellularization will be taking place, and based on the stage of the cellularization, so it will be converted into number of cells like this. This is a change which is taking place during the development of the endosperm. Finally, because of cellularization, the endosperm will be formed, which contains multicellular structure or multicellular nature or multicellular condition. But if you observe in examples like coconut, as we know that in the coconut two types of endosperms are present. One is called as liquid endosperm and other is called as a solid endosperm. Liquid endosperm means the coconut water or the coconut milk which we consider. So, here that is called as liquid endosperm, and cellularization will start from the periphery and gradually that will be converted into multicellular structure, that is we call it as a copra in coconut. So, likewise the amount of liquid endosperm and solid endosperm will be changing based on the cellularization which is taking place inside the endosperm. So, this is what the development or changes which are taking place in the endosperm of the different types of plant species. As we said that endosperm is a nutritive tissue which will provide nutrition to the developing embryo inside the seed. So, based on that one, two types are there. One is non-endospermic seeds and other is the endospermic seeds. Non-endospermic means by the time of seed maturity, if the embryo completely consumes the endosperm, that means endosperm is completely exhausted by the developing, utilized by the developing embryo. So, at the time of maturity there is no endosperm inside the seed. Such type of seeds are called as non-endospermic seeds. So, which can be seen in pea, beans, such type of examples. Whereas endospermic means the embryo cannot consume the entire endosperm which is present inside the seed. So, by the time of seed maturity some amount of endosperm is left out. Such type of seeds are called as endospermic seeds, which can be seen in coconut like examples. So, this is a deviation or types of endosperms that we can see based on the amount of endosperm that is left out in the matured seeds. There are some other type of conditions where perisperm is also being present in the seeds. So, the such type of seeds are called as perispermic seeds. So, actually nucellus which is present around the embryo sac, it is also acting as a nutritive tissue. In some seeds the nucellus is remnant of nucellus is present. That remnant of nucellus is called as perisperm, and those seeds which contains a perisperm are called as perispermic seeds. So here actual nucellus is also a nutritive tissue which is providing nutrition to the embryo sac and the structures present inside the embryo sac. So by the time of complete maturity of the seed, if some amount of nucellus is left out, that left out nucellus is called as a perisperm. So this type of structures can be seen in the coffee beans. And wherever the seeds are non-endospermic, in those type of seeds the perisperm is seen, whereas in the endospermic seeds perisperm is absent. So non-endospermic and perispermic seeds are present. So it is not compulsory that all non-endospermic seeds should be perispermic, but perisperm is present in those seeds where the endosperm is completely consumed. So these are the changes which are taking place during the development of the endosperm.